So for today, since we will see, I don't think so, but I wanted you guys to do these videos. Why? Because tomorrow, I think tomorrow, let me check. Yes, tomorrow in our class, we will be discussing so many bacteria in Sketchy. So I wanted you guys today to do the Staph aureus and the Staph epidermidis and the Staph saprophyticus. So today I can ask you, and the group A and the group B, so today I can ask you to do the rest of these. So tomorrow we will have more to cover basically. But today I don't expect you guys to know the Sketchy. I'm expecting that for tomorrow. But for those who did not prepare Sketchy for today, that means you have double the work for tomorrow. But you must do it, guys. Because if you don't know it, if you don't do it, like when we do our classes, you will be basically confused. There's a lot of concept you won't get. So you must do it. I sent a link with the Sketchy. I realized that many of you had a problem like installing it. Please know that you need to use a software called WinRAR. So the idea of WinRAR is to unzip your file because this file is basically compressed and you have to unzip it. So you can just come here, download this. After downloading it, install it. After installing it, you can just use it to open the folder. And when you use it to open the folder, you are practically done. So let me just send you a copy of this. Oh yeah. Yeah, and that is pretty much it. If you still have problems doing that, even with this, please contact me after the lecture. In 5 minutes, I will just call you and we can do it together to finish up with it, okay? And that's pretty much it. Good. So let's start our today lecture then. So today there is a few concepts we are going to discuss. We are going to speak about spore-forming bacteria, bacterial virulence factor, and some bacterial genetics. To be fair with you guys, this bacterial genetic thing is difficult. It's not easy, but the moment you get it, that's it. You are done. There is a few things that I want you to, stupid mnemonics that I made for a few of them. If you memorize them, it will be a piece of cake. And then we'll be speaking about what is endotoxin, what is exotoxin, and finally today we'll speak about bacterial toxins, which there are so many of them. So our day today will be full with toxins and genetics. Let's start with the spores. Spore-forming bacteria. First of all, what is a spore? A spore is a particle, basically, that contains genetic material of the bacteria. So you have the genetic material of the bacteria, and it's used for what? Survival. So these spores are used highly for survival. Well, for example, can some form of bacteria live above 100 degrees? Yes or no? Can bacteria live over 100 degrees? Yes, some of them can. Well, can some spores stay alive for thousands and thousands and thousands of years? Yes, they can. Can the spores stay in the soils? Yep. And not only tetanus, also anthrax. You know, maybe Yellowstone. There is this Yellowstone. It's basically a resort in the US, and this place have a volcano basically. And inside of that volcano, they were able to isolate some anthrax. You're like, wait a second, isn't volcano hot? It's super hot. And they still found anthrax. Why? It was in the form of spores. So can spores survive increased acidity or decreased acidity? Yes or no? Yes, they can. So anthrax basically badass. They can survive anything. So some gram-positive bacteria can form spores when nutrients are limited. It's basically their capsule to transmit to the next generation. They are like, okay, I'm dying. Let me make one last survival wish, in which they make like this little spore with a really thick coverage around it. Oh yeah, it's like a time capsule. And they leave it. After some times, it can reappear. This is kind of scary. Why? Because what is melting in the North Pole? Yep. There is a theory, actually, guys, about us humans that one of the causes of our extinction will be due to viruses and bacteria that are under the ice, basically. They are hiding under the ice in the North Pole and the South Pole. And when the ice basically melt, they will get out and they will say for us, hello. Well, that is not a good idea, you know? There is a high chance if these viruses will get released and they will be picked up from some humans, that can lead to our extinction. So yeah, one more problem with global warming. So let's go back here. So the spores lack metabolic activity. They are basically like seeds like tree seeds. Lack metabolic activity, and they are highly resistant to heat and chemicals. The core of them contains dibiclonic acid, okay, which is used for the heat resistance. And you must autoclave them. What is autoclave? I will show you in a second. It's basically a device that you use for heating so much by steam, by creating a really hot steam to kill the spores. You need to do it for 15 minutes over 121 degrees Celsius. This is not Fahrenheit, guys. So hydrogen peroxide and iodine-based agent are also sporicidal. So hydrogen peroxide, does anybody remember how hydrogen peroxide works? How does hydrogen peroxide work? It makes free radicals. Okay? So there is something here that is important. We can see here anthrax, B series, which is Bacillus cereus poisoning, which happens due to basically eating reheated rice. Yeah, reheated rice. And also Clostridium Battalionism, C. diff, and C. perfringens, and C. tetani. All of these have one thing in common. All of these guys are gram, positive or negative, these all are gram positive. So if I'm speaking about the reheated rice, it's in the B series. Please know about the reheated rice. It's mostly fried rice. So in some population, they do eat fried rice, and then they leave it a little bit cold. And when it becomes cold, what have the time to germinate? What will germinate when you leave it cold with B series inside of it? What will germinate? The spores, yeah? So the spores will germinate. When they germinate, they make you more B series. So this B series will be like, oh, finally. And you will come and be like, oh, nice food, let me eat. So you will eat, you will get really bad vomiting, diarrhea that lasts for 1 day and then go away. Doesn't sound that bad until you get it. So it's mostly, actually, to be fair, the most common symptoms of it is vomiting. Don't worry about this stuff now. We will study each of them alone. But the only thing that I wanted you guys to know, that there is something called the spores, and it's like a time capsule in which it transmitted to further generation. So what is an endospore? Well, bacteria can make spores in two ways. Either it will be inside of it or outside of it. If it will make it outside of it, it will be in the location, like in the area. But if it's inside of it, it's like you are like, okay, bacteria, I'm killing you. You shoot it, so it dies. But the endospore inside of it will be released and it will be not harmed. So it's kind of like a payback type of thing. So you kill the bacteria, but it has an endospore inside of it that gets released. Okay? So bacterial virulence factor. They promote evasion of the host immune response. Basically, all of the, not all, but some of the virulence factors of the bacteria, their main function is to run away from our immune system and kill our body. For example, in the Staph aureus, you will learn that there is something called protein A. This protein A is something like this. So let's imagine that this is the Staph aureus, and this is the protein A. Okay, this is a protein A on it. This is a must to know, guys. So if we imagine that this is the protein A, you know that an antibody have an Fc portion and a Fab portion. So next thing I'm going to say, this is particularly high yield, okay? So this is the protein A I just drew. Now I'm going to draw the antibody. I want you to know that it binds to the Fc portion of the antibody, okay? The Fc portion of the antibody. Well, if it binds to the Fc portion of the antibody, look what happens. If it binds to the Fc portion of the antibody, can this antibody go and bind something else? Or he's stuck? He cannot, he's stuck basically, yeah? I explained the difference between endospores and spores. So I said that spores are released outside of the cells. Endospores are within the bacteria. So if you destroy the bacteria, the endospores will become spores. Done. So if we speak about this area, you can see this antibody, it gets basically neutralized, yeah? By what? The protein A, which is located in what? Staph aureus, which prevents what? The IgG from exerting its function. What is the function of IgG? Opsonization. So the IgG cannot opsonize anymore, and if he cannot opsonize anymore, you will have really bad phagocytosis. So basically, this Staph aureus knew what to do and had this protein A, which neutralized the IgG. No opsonization. Remember what was opsonization? It was tagging. And no phagocytosis. Yep. That's all done by the Staph aureus. So the next thing I'm going to speak about is the Iga Protease. So it's an enzyme that cleaves IgAs. Can somebody remind me, please, where can I find IgA? Exactly. Yep, correct. It's the mucous membrane. It's the mucous membrane. So IgA is normally located on the mucous membrane. So if we, and the mucous membrane can be inside of your nose, inside of your respiratory tract. They can be inside of your GIT, the genitourinary tract as well. So these IgA proteases basically cleaves and destroy the IgA. Guys, just really quick, can somebody tell me, was an IgA antibody, was it a monomer or a dimer? Well, yep, that's a full answer. Good. So it's normally monomer when it's inside, but when it becomes secretory, it becomes a dimer. What is the particle that is needed to connect two IgAs together to make a dimer? Exactly, it's the J chain. You must know about the J chain. The J chain connect the two particles of IgA to make a secretory IgA. Excellent. Oh yeah. So enzyme that cleaves IgA, allowing bacteria to adhere and colonize to the mucous membrane. Why? Because this IgA protease comes here and he's like, well, let me come and do a trick. No. So he destroyed the antibodies basically. So when he destroyed the antibody, it becomes no antibodies. So the bacteria will be like, oh, nobody to meet me. Okay, I will consider myself at home. So the bacteria, which is like the strep pneumonia, H. influenza, and Neisseria, all will come in. By the way, these bacteria mentioned here, what they have in common, guys? Neisseria, H. influenza, type B, and Strep pneumo. All of them have one thing in common. They all have capsule and they all are IgA protease positive. Okay, so these can come and adhere to the mucous membrane and they can lead to infection. Okay, so the last thing I want you to know, guys, is the M protein. So this M protein is quite interesting and it's really important, and we spoke about it a little bit in cardiology, but now I'm going to expand on that. So if we imagine that we have a bacterial cell, this is a bacterial cell. Let's imagine that this is a group A Streptococcus, beta hemolytic Streptococcus. I will tell you in the next class that it has something on it. It's called the M protein, the M protein. So this M protein is located on the walls of the bacteria. Of course, it has a function. What is the function of the M protein? It prevents phagocytosis. Well, basically, when the phagocytosis by macrophages comes to eat these cells, he sees the M protein. So this is the bacteria. She has a lot of M. And when the phagocytes come, which is the macrophage, he's like, oh, I see a delicious dish. Let me come and eat. So he sees this bacteria and he wanna eat it. You know what she tells him? How dare you? I have an M. I'm a mom. Don't eat me. You cannot eat me. I'm a mom. And the macrophage feels bad and he decides not to eat it. Why? Because she has an M. She's a mom. So I want you to know that the M protein, it prevents the phagocytosis. Why? Because she's a mom. Cannot eat a mom. Okay, good. She's a mom for what? Molecular mimicry. So it helped to prevent phagocytosis, which is expressed by the group A strep. Here it is. Sequence homology with the human Trobomyosin and Myosin. Let me tell you what is myosin and the Trobomyosin. If you remember, guys, we spoke about in cardiology about something called the Trobomyosin and Myosin. These will be explained further when we discuss the musculoskeletal system. But the general idea is like this. You have an actins and you have basically myosin. And these myosin and these actins, the myosin head binds to the actins and they lead to contraction. Okay? So what happens? This M protein mimics the myosin and the Trobomyosin. So they have similar structure to the myosin and the Trobomyosin. It's like, I'm a mom. I have M, which look like myosin. Yeah, it's like my cousin. It looks like an M. So why this is important? When you start making antibodies against the M proteins, well, phagocytosis did not work. So what your body will start wanna make? Antibodies. So he will make antibodies. Great. You make a lot of antibodies. And these antibodies are specific against what? You guessed it, the M protein. So they will be working against the M protein. This M protein looks exactly like what? It looks like your myosin in your heart, yeah? So this is actually could be the reason. I'm not telling it is the reason because it's unknown that why the M protein prevents phagocytosis. Maybe because the macrophage think that it's your own body and don't want to phagocytose your own body. Anyway, when you make these antibodies, they will come and let the war begin. Boom. They will destroy the bacterial cells. One more, boom, destroy the bacterial cells. Okay, but you made a lot of antibodies that were enough for everything. Where will go the other antibodies that were made against the M protein? If you destroy the bacteria, you still have antibodies. What you will attack? What will the antibodies against the M protein that highly likely resemble the myosin attack? They will go and attack the myosin. Where is myosin located? Inside of the heart. Excellent job. So when they attack the heart and actually the muscles, you are correct. So when they attack the human cells, We call that molecular mimicry, okay? So possibly underlies the autoimmune response seen in acute rheumatic fever. Okay, so I hope guys you understood this little story about M proteins and basically the mimicry. Yeah? Okay, good. Nice. We are done with this. So, the next thing is kind of difficult. I cannot lie at you. This is a bit difficult. This is super easy. This where the fun start coming, and this is all easy until we reach virology. I know that some of you might suffer a little bit in virology in the beginning, but then it becomes easy again and a lot of fun. Microbiology is a little bit lengthy, but it's not a difficult subject, guys. So, but let's finish this one hard topic that exists in it. So, I will try to go with it a little bit at a time because I know how it goes. So, bacterial genetics. First of all, I don't want you to get confused. We have two pages in first aid responsible for genetics of microorganisms. The first one is bacterial genetics, it's here. The other one will be viral genetics. It's in a totally different page. It's a totally different things. They are not related to each other, okay? So that viral genetics is recombination, reassortment, complementation, and phenotypic mixing. That's different. Today we are speaking only about bacteria. We don't care about viruses. So, yep. But I need to warn you, I will be giving you tricky questions today. So, starting with the transformation. What is transformation? Well, basically you had a bacteria. Well, you have a bacteria right now. And this bacteria is there and she's like, well, yeah, what are you doing? I'm bored, I'm doing nothing. Okay, there is a piece of a free DNA. Would you like to have it? And she's like, yeah, sure, give it to me. I want to internalize it. He's like, wait a second, don't you want to check what it is? She's like, no way, I'm too stupid for this. I don't have a brain, just give it to me. That is the process of the transformation. Basically, bacteria internalize pieces of DNA that they have no idea where they came from. Okay? So, they say, competent bacteria can bind and import short pieces of environmental naked bacterial chromosomal DNA. So let's say that in this environment there was bacteria previously in this area, but they died. Yeah? See? The X's in the eyes always mean death. Okay? They died. After a cell dies, what happens to the cell wall? Can somebody tell me what happens to the cell wall after the cell dies? What happens to the cell wall when the cell dies? Can you hear me, guys? Well, when the cell dies, the cell wall basically bursts and get destroyed. So the internal material gets released outside. Yes, you can see here, a massacre just happened. A lot of pieces were released into the area. From a lot of pieces of DNA will be released inside of the area of the dead cells. Yeah? These are the pieces that the healthy and competent bacteria take inside of it. Basically, it is like the dead cell is is like putting a message in the bottle and throwing it and waiting for somebody to pick it up. So, competent bacteria can bind and import short pieces of environmental naked bacterial chromosomal DNA from bacterial cell lysis. So previous cell dies, bacterial cells, and then the naked DNA makes it inside. So, as you can see here, we have a recipient cell. The naked bacteria is imported inside. So, the transfer and expression of a newly transferred genes is called the transformation. So, you got these pieces of DNA. They are inside of the cell. What is the big deal? Well, you start cutting it, okay? After cutting it, you recombinate it. After recombinate it, you put it inside of the genome of the bacteria. Guys, you notice something different about the DNA of the bacteria. How does it look like? Is it linear or circular? Circular. And what organelles inside of our body has a circular DNA? What organelles inside of our cells have circular DNA? It is the mitochondria, guys. The mitochondria does have, yes, excellent. The mitochondria does have circular DNA inside of it. Because there is a theory that the mitochondria used to be millions and millions of years ago, prokaryote that entered our body and they start living with us symbiotically, which means we give her the heat and the area to live in, and she gave us energy and help us do a lot of metabolic activity. So I hope you get the idea what is a transformation. So transformation is getting a naked DNA from the outside to the inside, incorporated within the DNA, yes, inside of the recipient DNA, of course. And after that, after it's inside of the DNA, it will be expressed. What does gene expression mean? It's the process of converting DNA into protein. Normally, DNA, RNA, and then a protein, yes? So, don't forget the process of conversion of DNA to RNA. How do we call that? DNA to RNA. How do we call that? Excellent. It's a trans what? Transcription, yeah? While RNA to protein, translation. But how do we call when we convert DNA to gene expression? I mean 2 steps at a time. How do we call it? Moving from DNA to a protein. Of course, you go through the 2 steps, but I mean, how do we call it when the DNA becomes a protein? We call it gene expression. This is important, gene expression, okay? So, the next thing is, after it will do gene expression. So the transfer and the expression of a newly transferred genes is called transformation. A features of many bacteria, especially Strep pneumonia, H. influenza type B, and Neisseria. Okay? Again, it's the shin. Well, where did we see these before? What these two did to us that we spoke about today? They had one more thing in common. They had one more thing in common. Do you guys remember what was the other thing that they had in common? Yeah? They were all IgA protease positive. Yeah? Which means that they used to destroy the IgA on the mucous membrane. Yeah? Good. So, this is really, really high yield, guys. Okay, so they write for us, if you add the deoxyribonuclease, it destroys the naked DNA, preventing the transformation. Let me explain this. Well, basically, if you have a culture in vitro, okay, and basically this is a Petri dish. That's what I'm trying to say. It's basically a dish. Let me make a better dish. This is a Petri dish. Inside of the dish, you have some bacteria, and you have also free particles of DNA. Normally, these can go in and make transformation in which they incorporate their DNA in the recipient DNA and can be expressed. But if you give deoxyribonuclease, which basically destroy the naked DNA, you will have destruction of the naked DNA. So if the naked DNA does not exist, can you have transformation? You cannot. Let me repeat this one more time. You have a Petri dish, yeah? Inside of it, you have the bacteria, and you have portions of DNA. Now you bring something called the oxyribonuclease, which destroy the DNA. So now inside of the cell, inside of the petri dish, you add the oxyribonuclease. Of course, he cannot make it inside of the cell. So he will destroy all of the free DNA outside of the cell. So this DNA cannot make it inside, so you cannot have a transformation anymore. Okay? Kind of cool. Good. So the next thing is regarding, let's see, preventing transformation. So that's it about the transformation. Not that difficult of a topic. I know that it can get confusing. The thing about these, it's that they can get confusing. Yeah, that's the only problem with them. Yeah, so you just need to put one thing about each one that we'll never forget. For example, this one is naked. Yeah, like in first aid, I checked it out, maybe first aid 2012. It had a mnemonic that it was deleted, maybe because of sexism or something like that. They said like a naked trans, okay? A naked trans separate his DNA. But I think they canceled that because of sexism or something like that. But if it works for you, use that mnemonic, a naked trans. Okay. Oh, no. It was something about a naked trans transform himself, something like that. I can bring you the mnemonic. It's kind of interesting, but it helps you to memorize. That's the point. You need to, the problem between these that they sound alike. Transformation, conjugation, transduction, they sound the same. So you need to find something in each one to be able to differentiate them. Because on the exam, the question is super easy. They tell you there is a naked DNA that has inserted inside of the cell and was expressed. Now you know it. It's a transformation, no problem. But when they put you transformation, conjugation, transduction, you get lost. But when you correlate the word naked DNA with something, you can get it. Okay, now we'll speak about conjugation, the first type before the break, the second type after the break. So let's start with the F positive and the F negative. First of all, what is this F? We are speaking about what? We are speaking about the plasmid. So what is a plasmid? A plasmid, if this is a bacterial cell, it has circular what? DNA. It also has a minor DNA located in it, inside of an organelle called the plasmid. It's important. The importance of the plasmid, that it has some genetic material inside of it. And if they mention, some cells has basically genetic material outside of the bacterial DNA itself, think plasmid. So that plasmid main function is actually to do what? Is to store some information regarding the survival, like antibiotic resistance, like, for example, making some enzymes against the antibiotics. Yeah? So the plasmid particles are super important for survival of the bacteria. Let me send you a picture of the plasmid. Okay, so the next thing, so an F positive plasmid contains genes required for the sex pilus and for the conjugation. So let's speak about conjugation. What is conjugation? When you conjugate two things together, you just attach them to each other. Yeah? So you say, let's conjugate something. So it's like, so somebody wrote this. Plasmid is a DNA outside of the DNA, inside of the bacteria. Exactly correct. It's a DNA outside of the DNA. Okay? Absolutely correct. You need also to know it's a circular double-stranded DNA. Okay? So epiplasmid contains genes required for sex pilus and conjugation. So they make the sex pilus. Can you see the sex pilus? What is its function? Well, this pilus basically connects these two. So yeah, they just got married. Let's celebrate. So what is this F positive and F negative? I told you that the F says plasmid, but what about positive and negative? Positive donate, negative receive. Positive is the male, negative is the female. That's it. So, okay, bacteria without the plasmid is termed F negative. So this does not have a plasmid, this have a plasmid. So you logically, who will give the plasmid to the other one? The positive will give the negative, yes? So 6 pillows on the F positive bacterium contacts with the F negative bacterium. Okay, we have a connection, excellent. One strand from the plasmid, not 2 strands, a single strand of the plasmid passes through the, what is this? 6 pillows to the other bacterial cells. So they write, a single strand of a plasmid DNA is transferred against the conjugal bridge or the mating bridge or the bellows, if you want to call it a bellows. So they pass it through it and they are formed in this area. Now notice what happens. Now this is a step they don't mention here. So now we have 2 cells. They have their circular DNA, their normal circular DNA, and now the first cell only have one strand of the DNA because it has donated the other one here. Yes, don't forget that we have this bellows. Yeah, we still have the bellows. Good. So we have a bellows, we have the connection. Good. So what will happen next? See this connection? We'll destroy it. We don't need it anymore. What about this DNA? Well, this DNA will create a strand next to it. It will duplicate a strand. What about this one? This will duplicate a strand as well. So wait a second. How do we call a bacteria with a strand with a plasmid in it? Is it F positive or F negative? A bacteria with a plasmid in it. Okay, exactly. This is what you get asked about, guys. So a bacteria that gets a plasmid exactly becomes positive as well. Good. So it becomes, it's not good for us, but good for the bacteria. It's become positive as well. So what can it do? It will transfer it to another bacteria that does not have a plasmid. Yeah? And this is exactly how bacteria transfer the antibiotic resistance between each other. Okay? So it's just like a secret message. They are like, okay guys, so these U-boats work with this Shifra and you need to know exactly how to deal with them. You need to make beta-lactamases. You need to create pores to kick them outside of you. You need to make this, that, all of that. All of the genetic knowledge required for the antibiotic resistance is stored inside of the plasmid. And by the process of positive conjugation, you can transfer the whole idea of how to become resistant against an antibiotic using this little bellows. Okay? And I hope this conjugation process made sense. Just remember, you always need a bellows in case of conjugation. Okay? You cannot have mating without a bellows. Okay, good. So now we'll take a little break for 10 minutes, then come back, speak about this concept, and speak about the transduction and the transposition. So see you guys in 10 minutes. Okay, everybody, welcome back. So I'm just going to send a link I got from one of the members of our group. It basically contains all of the microbiology sketchy. So it's a really good idea to do them, guys, from here. You don't need any program to open them up. You can just like enter the link here like this. Wait a second. For it to open, you can go directly to bacteriology, gram-positive cocci, and this is the Staph aureus. So for tomorrow, guys, I want you to do all of these actually. For tomorrow, you must finish all of these 5 videos, okay? So this is your task. You can watch them online. You also can download them if you want. So in the same time, after you finish these 5 videos, for those who done these 5 videos for today, because that was my request for today, please, after you finish with them, do 1, 2, 3, 4 videos. Sorry, 1, 2, 3, 4, 5. All of the Clostridia basically, and the anthrax, okay? So until this video, you must do this. As you can see, guys, they are not that long. 5 minutes, 8 minutes, 7 minutes, 6 minutes, 9 minutes. So you can finish practically all of this in a matter of a couple of hours. That's it. So, yep. So now we can move on. If you still have any technical difficulty, please let me know. We have to start this. Let me just open the chat on my phone. Yes, please do this. Today we had to watch only 2 videos, no? So these 2 videos are... Oh, okay, I thought I wrote one more. Okay, my mistake. If I did not write that, it's on me. Let me check something. Sounds suspicious. Okay, well, for tomorrow, I want you to do the following. Please finish up all of the videos from the gram-positive cocci, and then go to the gram-positive bacilli. Finish the first 3 to 5. I will not push you guys. As much as you can, finish. It's for your own benefit. I have to say, these videos are a lot of fun. You can enjoy them. Really, watch them to enjoy them. And yeah, basically, they are cartoons. And we will discuss them. So, yep. Okay. Now we move on. We are going to speak now about the rest of the procedure of the conjugation. So, there is something called HFR, which is high frequency recombination. When you just hear the word, you are like, what the hell is that? It makes no sense. I agree, it does not. But what does the well, basically F negative mean? F negative, you know, guys. What does F negative mean? A cell that is F negative, what does it mean? Yes, it gets a plasmid, which means it lacks a plasmid. It lacks a plasmid. So, exactly, no plasmid inside of it. So an F-positive plasmid can become incorporated within a bacterial chromosomal DNA. You're like, wait a sec. Are you telling me that the plasmid goes inside of a chromosomal DNA of a bacteria? Yes. So remember, guys, inside of a bacteria, we had 2 DNAs. The first one was the bacterial DNA. I'm drawing it in blue. The next one was the plasmid. I'm making it in red. Okay? If they tell you about like something like this, they tell you about a cell that has this. Basically, let's try to make a bacterial cell. And inside of a bacterial cell, we have what? We have this DNA, bacterial DNA, and there is also a plasmid. But the plasmid inside of this cell does not exist. Instead, they will conjugate. They will connect to each other. Yeah? Multiple things can happen, but what I want you to know that a plasmid can come from here to here. You are like, yeah, we know. One layer of the plasmid will come here and it will just sit here. No, that was in the first case. Now the situation is different. Now the plasmid will come from here and it will go to bind to the chromosome itself. You are like, wait. Are you telling me that the plasmid takes a piece of itself and incorporated and conjugated to the chromosome, to the DNA itself of the bacteria? This is exactly what I'm saying. Some pieces of the plasmid will be incorporated within the DNA of the bacteria. So what they write here, An F-positive plasmid can become incorporated into bacterial chromosomal DNA, okay? So, and when this happens, we call it, well, this cell here, we call it high frequency recombination. Why? Because we took a plasmid and we put it inside of an F negative cell inside of the chromosome. Okay, so now it changed its name to an HFR, which is high frequency recombination. Okay, cell, high frequency recombination cell. So we get the plasmid and we put it inside of the DNA of the bacteria itself, not inside of the plasmid. Okay, you are like, well, I'm fine with this. So you're telling me that you got a plasmid, see guys, the plasmid, and you moved it to an F negative cell, but instead of it being like going and forming a new plasmid, it was incorporated inside of the genome of the bacteria, as you can see here. It's exactly the same. And after that, now you have this HFR, or also called high frequency recombination cell, which is basically totally a new cell. So what's up? I got this. Did you get this first point, guys? This is the first step. We get an F positive cell. We transfer the plasmid to an F negative cell. And when it goes to the F negative cell, the plasmid, instead of making a plasmid in the cytoplasm as it done in the first case, now it's incorporating the DNA inside of the bacterial DNA. Okay? Directly. And it's changing its name to something called high frequency recombination cell. Okay, now comes one more innocent F negative cell. Here it is. It will have its job in a moment. So they write, a transfer of a leading part of a plasmid and a few flanking chromosome gene. High frequency recombination might integrate some of those bacterial genes. Recipient cells remain F negative, but now may have a new bacterial genes. So let's try to understand what they speak about. Well, basically this HFR cells, they have the original genes or not? Do they have the original genes or not? Tell me, yes or no? Do HFR positive cells have the original genes? Yes, they do. They do have the original genes. Do they have the plasmid genes? Do they have the plasmid genes? Yes, they do. They have both. So, is it in one strand or two strands these genes located? Both of them. Yes, in both of them there is strands that are located. Absolutely correct. I agree. So let's see the importance of this. So it's in both strands. So what will happen that one of these two strands will be cut, which is basically like the leading strand and the lagging strand type of thing. So after the plasmid has incorporated into the bacterial DNA, there will be a transfer and replication of a part of it inside of the other cell. So look what happens here. You get one strand and now it's moving to the other cell. Well, what are you transferring exactly? You are transferring the original genome in addition to the plasmid genome together on a one strand. Yes? Okay, what is the big deal? Well, when you get this to the new cell, which is this one basically, what will happen? You will be able to infect larger portion. Yes? So, yes, exactly. So from the HFR cells to the F negative cells, now we have a one more form of conjugation happening. So conjugation happened first time, now conjugation happening another time. But this one is transferring, double mutated type of material, which contains both the plasmid genome, the bacterial genome, and it's moving to the new F negative cells. When this happens, what you will have? Well, always they don't draw the whole thing, so I must do it. So What will happen next step? See this, guys? It will duplicate and be created here, okay? So that will form something like this. Done. So you will still have the HFR cells. Why? Because the last strand will be remade. No problem. We have remade the last strand. What about the new one? Well, basically, this is a really long strand, don't you agree? Look how long this is. It's really long and it contains different genomes. So when you take it and put it inside of the new genome, the thing that will happen basically, that it will go and incorporate inside of the F negative cells, yeah? And now it's making the majority of the genome of the new cells. We call this a recombinant F negative cells, which will be really, really disturbed from the original cells. If you compare this cell to the original cells, it looks almost completely different, yes? Why? Because now it has some of the F positive cell genome. It has some of the plasmid genome, and it has some of its original genome. So it's totally recombinated. So they write a transfer of leading part of the plasmid and a new flanking chromosomal gene. What is that? So these are basically the pink, green, and the blue. Pink, green, and the blue. They are the what? They are the plasmid strand. But if we speak about the blue one, it's sorry, not the blue. This one, I don't know what color is this. I think it's brown. So the brown one is from the original cell, which is the lacking chromosomes. And they all go to the new cells. So high frequency recombination may integrate some of those bacterial gene. Okay? Recipient cells remain F negative, but now they may have a new bacterial genes. Guys, take a look at this. Does it resemble the original F negative cell? Yes or no? Does it resemble its genome the same as the F negative cell? Yes, it might make the bacteria resistance. It does not resemble it at all. And that's the idea. It can make your bacteria basically completely resistant to it. It can lead to other stuff as well. Not only the resistance of the bacteria that is important. It can make it resistant to the heat. Yeah? So, or to some enzymes, for example. So they write, recipient cells remain F negative but may have totally new bacterial gene. Okay. So let's take a look about the last procedure. So we spoke about the transformation, we spoke about conjugation, and now we are going to speak about a procedure called a transduction. The first thing I want you to know about the transduction, that it involves viruses. In a case of a transformation and conjugation, it was all bacterial to bacterial. We did not have any involvement of viruses. What I want you to know that whenever you hear bacteria plus viruses, think transduction, okay? So you hear the word bacteria, viruses infecting bacteria, leading to movement of genetic materials. Don't think a lot. It's a transduction. So you can see why students make mistake, because the transduction and the transformation sound kind of the same, yeah? So, okay. So let's take a look. Basically, I had a friend that used to say, oh, no, I told him, man, I keep confusing transduction with the transformation because of the name. He told me, no need to confuse them, because the transduction one, it has a duct in the name. I was like, what does that mean? He told me, well, take a look. The viruses here is making a duct to put the bacteria inside of the cell, to put the genome inside of the bacteria. I was like, yeah. He was like, so it's a transduction. So a transduction. So there is two ways of transduction. There is the generalized way and there is the specialized way. So the first one is a backing error. So first of all, you need a lytic phage. So please write. A phage here in this case is what? The lytic phage, it's a virus, okay? So are viruses alive or dead, guys? Are viruses alive or dead? They are dead. Good, they are not alive. So lytic phages infect the bacterium, leading to cleavage of bacterial DNA. So this is the bacterial DNA. Now, basically, we have this lytic phage has injected its nucleic material inside of the cell and led to lysis of some portion of the DNA. So after that, parts of the bacterial chromosomal DNA may become packaged in a phage capsid, and phage infects another bacterium transferring these genes. Let me explain. First of all, what is a capsid? A capsid is a structure that contains nucleic material inside of it. It's the packaging form of transferring the info. So a phage has a capsid, yeah? And it injects the nucleic material. Now, this nucleic material will have one thing. So it's not, and notice please, it's not added to the original what? Genome. It's not. You just cut it. After cutting it, you make a lot of capsid. Some of them with the bacterial genome, and some of them from the original viral genome, which led to what? Cleavage. Yeah? Okay. So now you have release of the new capsids, making new phages, and after that, some phages contain what? They contain bacterial genome. This bacterial genome will be injected to another bacteria. Yeah? If another bacteria gets this new bacterial genome, it will have incorporation of this portion into the cell genome. Guys, if this procedure happens, that means that after a generalized transduction, you can have in this cell, this one, a new characteristic of the cell, like for example, antibiotic resistance. To be fair with you guys, the main reason that they teach you the genetic of bacteria is because of the antibiotic resistance. So the last thing I want you to know is something about the specialized transduction. So when you hear the word transduction, you think about what? Transduction. There must be a, yes, viruses. You need to check for a duct. Yep. Who make the duct? The virus. So in this case, it's a little bit different from the first one. You will see why in a moment. So it's an excision event. So a lysogenic virus infects the bacterium, but this time the thing is a little bit different. Look, in the first time, you had a cleavage of the bacterial DNA, but you did not have incorporation of the bacterial DNA inside of it. But now you have incorporation of the bacterial DNA inside of the, you have incorporation of the viral DNA inside of the bacterial DNA. So that is the difference between generalized and the specialized form of a transduction. So after this lysogenic virus infects the bacterium, viral DNA will be incorporated into the bacterial chromosome, as you can see here. When phage DNA is excised, a flanking bacterial gene may be excised with it. If you take a look here, the capsid now contains a genome that has both the bacterial DNA and a portion of the viral DNA. So you have created a monster. You created basically two DNAs in the same structure. So they write, when the phage DNA is excised, flanking bacterial genes may be excised with it. DNA will be packaged into phage's capsid afterward and can infect other bacterium. As you can see, the new phages will be released, infect another bacterium, deposits the new material forward. It can occur in only 5 types of bacteria, I cannot tell you that, guys, you must know about these. You should know about these, but it's not you must know about these. It's not super high yield, but it's nice to know everything. So it's in a group E Streptococcus, the erythrogenic toxin. Also B for botulinism toxin, cholera toxin, diphtheria toxin, and the Shiga toxin. Basically, what are they trying to say? That these lysogenic phages are the reasons why these bacteria make their toxins. Okay? So that's it for specialized. For specialized, they help the bacteria make a specialized form of exotoxin. While in the generalized, you are changing the genes of the bacteria to make them more resistant. Okay, so the last thing is the transposition. So again, I told you guys, it's kind of complicated, the names. Transformation, transduction, transposition. So transformation, well, it was naked. The transduction, it had a duct. Transposition, well, it moves from one position to another position. So it's a jumping process involving a transposition. So it's a specialized segment of the DNA. What I want you to know that this transposition is actually motile. It moves all over the place, okay? So if you imagine that this is a transposition, he can be in the DNA, can go outside of the DNA, it can go around basically, okay? So it's a jumping process involving a transposon, which is a specialized segment of the DNA, which can copy and excise itself, okay? And then insert into the same DNA molecule or an unrelated DNA, like a plasmid or a chromosome. So basically you have the transposon here. He will decide that he want to cut himself outside of here and go to the plasmid, for example. So, or he want to go to another gene, yeah? So this plasmid, this transposon is kind of a tricky guy. He can go wherever he wants. So, which can copy and excise itself and then insert into the same DNA molecule or an unrelated DNA, like a plasmid or a chromosome. Critical in creating plasmid with multiple drug resistance and transfer across species line. So basically, we will learn about a form of Enterococcus. It's a type of bacteria, okay? This Enterococcus is resistant to vancomycin. And if they ask you about the mechanism of its resistance, it's the transposon transferring the idea and basically the code of the resistance to where? To the plasmid. Yes, they will mention a molecule that can cut itself from the DNA and move to the plasmid. That's a transposon. It happens in the transposition, and it can lead to a bacterial resistance. And we are done with the bacterial genetics. I cannot say that it's easy. It's not. It's difficult. It's one of the difficult topics, guys. But if you do it twice or three times, you understand the ideas, you correlate one word with one name, you are done. Naked, transformation. Conjugation, the 6 pillars. Transduction, duct with the phages. Transposon, it keeps jumping, yeah? From the DNA to the plasmid. Okay, now let's speak about endo and exotoxin. So exotoxin is in both gram-positive and gram-negative bacteria. It is secreted from the cell, okay? So it's produced inside of the cell. As you can see, this is a bacterial cell. It makes exotoxin, which goes to the host cells and affect it, yeah? Leading to downstream cellular reaction. So it is secreted from the cell. Its chemistry, it's basically just a polypeptide. It's a toxin. The location of the gene, it can be the plasmid or the bacteriophage. I have a question. Guys, what is a bacteriophage? What is a bacteriophage? Exactly, bacterial virus. Yep, it's a virus. Okay, so in which of the genetic stuff for the bacteria we mentioned a bacteriophage? Yeah, the one with the duct, exactly, the transduction. Yep. So the exotoxin are highly fatal. The fatal dose is on the order of 1 microgram. So that's really small amount. Yeah. So of dose, I mean, which means basically you need too little of that toxin to kill the patient. Yep. So it's highly fatal. Clinical effect, various effect, depending on the type of exotoxin we're speaking about. Look at these two pages. These two pages just speak with you about different exotoxin and how they work. So, the clinical effect varies. Mode of action also varies. We'll speak about all of them. The antigenicity. So, they can make a high titers antibodies called antitoxins, which can be basically counteract the function of the toxin, which is a good thing. So do we have vaccination against these? Yes, we do. We have toxoids. And are they heat stable or heat labile? Well, basically, they are labile. They can be destroyed by the heat. Destroyed rapidly at 60 degrees, except some of them, like the staphylococcal enterotoxin and the E. coli heat stable toxin. Since they are heat stable, they will not be destroyed. Only these two. Okay, so typical diseases include tetanus, botulism, diphtheria, and cholera. We'll speak about each of these exotoxin and how it works exactly. So, the endotoxin, on the other hand, they are not released. They are just located as a portion of the gram-negative bacteria. What is the analog for the lipopolysaccharide in the gram-positive bacteria? Does anybody remember about this? Analog for lipopolysaccharide in the gram-positive bacteria. That leads to fever and all of the good stuff. Excellent. Lipotocoic acid. Yep. So the endotoxin source outer cell membrane of most gram-negative bacteria. They are secreted from the... Do they get secreted from the cell? The answer is no. Why? Because they are a part of the cell. So when do they get released? When the cell is destroyed. Yeah? So location of the genes, they are from bacterial chromosome, which means from 100 million years ago and now it is the exact same lipopolysaccharide. So the toxicity is low, which means the fetal dose, it can be up to hundreds of micrograms. And the clinical feature, they can lead to fever and shock and DIC. Can somebody tell me how they lead to hypotension, endotoxins, lipopolysaccharide? How can lipopolysaccharide lead to hypotension? Agree, septic shock, but how exactly they vasodilate the vessel? They activate nitric oxide. Exactly, they activate nitric oxide. Yep. They activate it by binding to the endothelial cells or by binding to the smooth muscle cells. It's important to know this one. Yep, it's important, but the answer is wrong. It's actually the smooth muscle cell. So that's super important because they do go to that little details. I think personally it's stupid, but you need to know about it. So bacterial lipopolysaccharide can make nitric oxide after the lipopolysaccharide binds to the smooth muscle cell. Okay? Okay, so these interleukins can also lead to more TNF-alpha, interleukin 1 and 6. So they are poorly antigenic, which means you cannot develop immunity against them. And no toxoid forms and no vaccine is available. Yeah? So they are stable at 100 degrees Celsius for 1 hour, which means they suck. You cannot destroy them easily. Typical disease in case of meningococcemia or sepsis by gram-negative bacteria, especially E. coli. Yeah? As you can see, the endotoxin is only released when the cell is destroyed. Okay, good. So let's take one more break and come back in 10 minutes to finish up all of these toxins we have left. So see you guys in 10 minutes. Let's see. Okay, so now we are going to speak about exactly how some exotoxin work. Can you see my screen, everybody? Just write me yes or no. Okay, good. So bacteria with exotoxin. So some bacteria inhibit the protein synthesis. If you don't have a protein synthesis, can you have productions of enzymes? Yes or no? No enzymes. Yep. If you do not have enzymes, will our cells be able to live or they will die? With no enzyme, death will occur. So inhibit protein synthesis. All of these inhibit the protein synthesis, but the mechanism, the exact mechanism, is a little bit different. So for the Corynebacterium diphtheria, which produce the diphtheria toxin, and the Pseudomonas aeruginosa, which produce, the Pseudomonas aeruginosa exotoxin, both of them lead to inactivation of something called elongation factor 2. So if you want to understand what is the elongation factor 2, this is one elongation factor that we are going to speak about in biochem. In biochem, I'm going to explain for you some points regarding how the processes of transcription and the translation and the protein synthesis occur. So in protein synthesis, I'm going to tell you something about elongation factors, okay? These elongation factors basically facilitate the formation of proteins. If you don't have normal elongation factors, you will not be able to have normal protein production. With no normal protein production, you will not have normal enzymes. With no enzymes, you will have death of the cell. Okay? So, so you can see the elongation factors, and basically without them, you will not have a protein synthesis. And yep, so let's take a look at this. We saw it down there. And please know about the elongation factor, and without it, there is impossibility of it making basically the process of translation. No translation, no proteins. No proteins, the cells is doomed for death. I want you to notice the little little A's is up there. What are these? This is basically writing that it's an AB toxin, which is the A does something and the B does something. The A is the active particle and the B binds, okay? So 2-component toxin, or 3 for anthrax, with B being the binding and it triggers the uptake endocytosis, while the A is the active component. The A active component are usually ADP ribosyl transferase. Other have enzymatic activity as listed in the chart. So let's take a look what is listed in the chart. So when we say that it inactivates the elongation factor and it will lead to less, which process? Translation. Less translation, inhibition of the protein synthesis. Please know that the ribosome, which is the organelle that normally makes our protein, is composed from 60S and 40S subunits. So what is this S? Actually, it's not a unit of weight. It's a unit of centrifuge. It's a unit of density. So when you put it on the centrifuge, you will be able to see these particles at the 60 spins per second, basically. And inactive 60S ribosome by removing adenine from the rRNA. So that's removing the adenine from the rRNA, which happens in which toxins? The Shigella and enterohemorrhagic E. coli, which also called, in case of enterohemorrhagic E. coli, the Shiga-like toxin. Please write a note here, Shiga-like toxin. So, the Shiga toxin or Shiga-like toxin can lead to destruction of endothelial cells. So let's take a look at these toxins one at a time and see what they cause. The diphtheria toxin led to the death of the cell by destroying the protein synthesis, which can lead to pharyngitis with the pseudomembrane making in the throats. Also severe lymphadenopathy called the bull's neck and eventually myocarditis. I want to just show you really quick these pseudomembranes because they do show you a picture of it. So if you take a look at the diphtheria, I hope you can appreciate this shape of this grayish membrane in the back of the throat. Guys, I have a question. Why we don't see it often anymore? Why we don't see it as often? Yep, because of the vaccination. Good. Remember the DTB vaccine, yeah? So that's one thing. I also want to show you. Oh yeah. You can see how big are their neck. It's ridiculously big. We call it the bull's neck, yeah? And if they ask you why it's big, it's just lymphadenopathy. They also can have myocarditis. Remember, myocarditis presents with acute heart failure symptoms. Okay, that was diphtheria. What about Pseudomonas aeruginosa? It can lead to host cell death. Well, for Pseudomonas aeruginosa, I just want to show you this skin necrosis that it can lead to. Oh yeah. Canthema gangriosa. So here you go. It can lead to a necrosis of the tissues inside of the skin. And if we speak about, so that was about the Pseudomonas aeruginosa, it kills the host cell and it can lead basically to necrosis. If we speak about Shiga toxin and Shiga-like toxin from the EHEC or enterohemorrhagic E. coli, these will lead to destruction of endothelial cells, okay? We can have Pseudomonas in contaminated water, sure thing we can. But since our immune system is good, it's less likely to get that type of infection. What you will get most likely is something like this. It's called hot top folliculitis, really common. If you see it, it's common in real life, like you see it in real patient, and also it's really common for the USMLEs. They will come to you with all of their body having so many little spots and it's really itchy, yeah? And you will take a look at it and be like, shit, what this patient has. He has the hot top folliculitis, yeah? But this is much benign condition than this one. This one you have to drain it, yeah? While this one is the hot top folliculitis, you just give him oral antibiotic and topical antibiotic and it goes away. So these toxins lead to destruction, in case of Shiga toxin and Shiga-like toxin, they lead to destruction of endothelial cells and epithelial cells, which can destroy the GI mucosa leading to dysentery. Please make a note. Dysentery means diarrhea plus blood. So dysentery is diarrhea plus blood. I hope somebody can write it in the chat. Dysentery is diarrhea plus blood. So enhances the cytokine release. So yeah, this Shiga and Shiga toxin and Shiga-like toxin will enhance cytokine release. So that can lead to HUS, hemolytic uremic syndrome basically, which can be due to EHEC, which is enterohemorrhagic E. coli. And also, this EHEC, it has a subtype called 10157H7. You need to know that. but it's good since there is not another other one that you have to know. So the moment you see this with Shigella-like toxin, you know it. So unlike Shigella, EHEC does not invade the host cells. So I want to show you some stuff, guys. So to start with, I want to tell you exactly the whole mechanism of the Shiga toxin. So inside of the blood vessel, in the kidney, for example, you have endothelial cells. These endothelial cells can be destroyed by the toxin, yeah? What is under the endothelial cells? You have this substance, collagen, yeah? Collagen will be bound to von Willebrand factor. Yes, in the basement membrane, basically, the collagen. And that will bind to von Willebrand factor, which will then bind to platelets, yeah? This will lead to formation of something called microthrombi. This microthrombi will basically lead to something called shear cells, which is basically when this RBC, when this little RBC, let me make one more RBC here. When this RBC, it's blue, but it's okay, comes here, it will be sheared by the microthrombi, which lead to formation of something called schistocyte, which looks like this. So schistocyte happens due to shearing, yeah? So schistocytes. This is important. Take a look at the schistocyte. Can you see how it's sheared? And the shearing of it happens because of the microthrombi, yeah? They also like to call it helmet cells. Helmet cells. So, okay, so the destruction of the endothelium can precipitate to what? It can precipitate to this patient developing the kidney failure, which is basically the enterohemorrhagic E. coli. Okay, so that was the first type of toxin, inhibiting the protein synthesis. We spoke about Corynebacterium diphtheria leading to the pseudomembrane in the back of the throat. We spoke about the Pseudomonas aeruginosa leading to the skin infections. We spoke about the Shigella and the EHEC and how they led to dysentery, which is diarrhea with blood. And also we spoke about how they led to kidney failure and formation of schistocyte. Now we speak about organisms that increase the production of a fluid. So the next one is Enterotoxic E. coli, which will be really useful for you guys when you do the sketchy. It will show you exactly and remind you about it. So it has a heat label toxin and a heat stable toxin. The heat label toxin, back in my time we used to say like something like that, stable on the ground type of thing. So this is labile in the air, stable on the ground, something like this. You will see what I mean in a second. So stable on the ground, the heat stable toxin work by a cyclic GMP. While labile in the air, it means it works by cyclic AMP. So actually this mnemonic does work because it's been like 1.5 years and I constantly use it, never forgot it. So heat labile toxin, they lead to overactivation of adenylate cyclase, which lead to chloride secretion into the gut. Guys, can somebody tell me the table salt is made out of what? Table salt. I mean the salt, the type of salt we eat. We put iodine in it, but it's not made of iodine. It's made out of sodium chloride, yeah, NaCl, yeah? So it's made out of sodium chloride. Okay, so chloride like sodium, that's what I wanted to reach. So when a chloride is more inside of your gut, due to the activation of the adenylate cyclase, you will have more sodium inside of your gut. What sodium likes? Which substance does the sodium like? Water, exactly. So these chloride channels right here are coupled into a cyclic AMP, yes? So when there is overactivation of this cyclic AMP, this chloride will be released into the lumen of the intestine. So when a chloride is here, what he will suck from the lumen to the lumen as well? Sodium. So when the sodium is sucked here, what will be sucked there? As you said, guys, correctly, it was water. Yeah, H2O. So we have water, H2O. Okay, more water here. So what will happen to the patient? When he has a ridiculous amount of water inside of his lumen of his intestine. You guess it. Exactly. Watery diarrhea. Yep. So it's watery diarrhea. Is there any destruction of the walls, guys? Is there any destruction of the walls, or the walls are intact? The walls are intact. Excellent. So since the walls are intact, can you have blood? You cannot. Exactly, no blood. So it's a watery diarrhea, not a basically bloody diarrhea. So watery diarrhea, labile in the air, it's the adenylate cyclase, and stable in the ground, which is the guanylate cyclase. Okay, so if we speak about the heat stable toxin, it overactivates the cyclic GMB, which decreases the reabsorption of sodium chloride and H2O in the gut. So yeah, these guys will not allow the reabsorption of the sodium chloride in the gut. Well, guys, which portion of the intestine most likely reabsorbs salts and minerals? It's the colon, guys. It's the colon, okay? Normally the colon reabsorb sodium and chloride. Why? To make the feces much more solid. Well, wait a second. You know, guys, somebody that has a colectomy, like they have no colon, they get an ileostomy bag, which is basically we get their ileum and we connected directly to the abdomen. I have a question. Since I told you guys that the sucking of the fluid from the feces make them solid, if somebody does not have a colon like this guy, and instead he has a bag like this bag instead, can you tell me please, will the feces inside of this bag be solid or liquid? Here is the thing. No sucking of the water. The sucking of the water happens where? In the colon. The patient does not have a colon. So, take a look. You cannot believe it. It looks like this. And actually it looks more whitish to be true. I'm sorry guys, but I want to show you, basically feces of an ileostomy bag. So basically, yep, that's basically how it looks like. They have their feces in a liquid fashion. They cannot be watery since, they cannot be solid. They are just liquid. Please advise your patient to always clean their bag, because the last thing you want is it to get infected. You don't want that, okay? So, so it's loose. Okay, good. So anyway, I was telling you about this just to remember that the process of reabsorbing the sodium chloride happen at the colon. So using the cyclic GMB, which is stable on the ground, a heat stable toxin, it will not allow the reabsorption. So more sodium chloride inside of the colon. That will suck what into it? If you have more sodium chloride inside of the lumen, what will happen? What will the sodium chloride suck? Water. Yes, there will be more water in the colon. So that leads to which type of diarrhea? Watery diarrhea. Yes. No destruction of the wall though. No blood. No destruction of the wall means no blood. So bacteria that increases the cyclic AMB include cholera, anthrax, Pertussis, E. coli, and each one of them does something totally different. For example, the cholera lead to diarrhea. The anthrax lead to edema of the wound. The pertussis lead to edema of the airways. The E. coli lead to diarrhea. As you see, each of one of them use the increased cyclic AMP for totally different function, and we'll be explaining it when we discuss each bacteria at a time. So the next one, Bacillus Anthrax. Guys, does anthrax make spores? Yes or no? Okay, it does. Well, yeah. So I wanted to tell you a story, guys. It's important for the USMLE. If you get a case in your clinic with anthrax, what is the first thing to do? If you are working in the hospital and you get a case of anthrax, no, you need to report it. That's the first thing to do. This patient is dead. The patient you have has 99% chance of dying. But if you speak, I mean like pulmonary anthrax. You need to protect yourself. That's true. That's primary thing. But you need to report it. It's one of the reportable disease. You must say that, okay, I got a case of this. You go tell the CDC right away. Don't wait. You need to report for the CDC. Because why you are reporting to the CDC? Do you remember where we normally found anthrax? Where we found the anthrax normally? Exactly, it's used as a biological terrorism agent, which means like if you have a case of it, probably there is more cases. Has it ever been used? It was. Well, was it used in the previous 20 years? It was. So it was used after 9/11 as a I don't want to get political, but basically after 9/11 there was some attacks, sending of letters, and inside of the letters they put basically the what? The spores, yeah? The spores of anthrax could live inside of the letter for basically months until they reach the US. Somebody opens it, inhales the spores, get pulmonary anthrax, and die. Did people die from this? The answer is yes. Many people died from this. I think like 20 or 30 people to be exact. So it's used for bioterrorism. If you get a case of it, you must report it. So Bacillus anthrax. It mimics adenylate cyclase. Again, as you can see, more cyclic AMP, which can lead to the characteristic adematous border of the black scar. Let me show you this. Please, no. You have two types of anthrax. You have skin anthrax and pulmonary anthrax. The skin anthrax you can get like from the soil itself. Like for example, if you work with wool, yeah? Like with sheeps and stuff like that, you can get anthrax. But the pulmonary anthrax, this is only bioterrorism. So take a look at this wound. Yeah, this is a perfect one. Oh, I was thinking why the hell it's so perfect, because it's Photoshop. Okay, look in the middle here. It's black in the middle and red at the rim, yeah? So that will be important in a second. Take a look at this lady. It's black in the middle and at the rim it is red. The black is the lethal toxin. Red is it due to edema? It's due to high cyclic AMP. Okay? Okay, so let's move on. We have likely responsible for the characteristic adematous border of the black scar in continuous anthrax. The next one is Vibrio cholera. Cholera toxins lead to overstimulation of adenylate cyclase by permanently activating the GS, which can lead to a lot of chloride being thrown inside of the GI tract, which can lead to really bad diarrhea. Since I really like you guys, I'm going to show you a video of somebody with cholera. Guys, the previous treatment back in the time was to actually put a bed with a hole in it so they can have basically this diarrhea without basically having to go to the bathroom. I know that it's really disgusting, but take a look here. You know, guys, when you put water inside of, when you put rice in water, it gives this liquid that looks interesting. It looks like this, yeah? This water. Yep, that's exactly how the diarrhea looks like. Enjoy. So what is the treatment of this, guys? What is the treatment of cholera? Oh yeah, I was speaking about this. What is the treatment of cholera? Rehydration, oral rehydration therapy. Let's say that you are in a country that does not have normal rehydration therapy. How to make it at home? You bring learn it. You need to bring 1 liter of water, add 9 little spoons of sugar and half of a teaspoon of salt. Okay? Mix it together, give it to the patient, drink it, drink it, drink, repeat. Or Sprite. Actually, that's true. That's really true. Okay, good. Cholera is horrible. It's infectious. You can get it if you eat the toxin. How can you eat the toxin? Fecal, oral. Use your imagination. So, the other ways that toxins can kill us is by inhibiting the phagocytes. Phagocytic activities. For example, in case of Bordetella pertussis, we can have basically what? In Bordetella pertussis, we can have inactivation of the inhibitory subunit of the GI, which is an indirect way is telling us that we have more cyclic AMP, which lead to what? The phagocytes not being able to be active. Somebody explained it to me like this. He was like, well, you know, when you have more cyclic AMP, you will have more edema around the cell. I was like, yeah. So he was like, well, let's imagine that this is the pertussis, yeah? It makes a lot of cyclic AMP, so there will be a lot of edema around the cell. Like, okay, so what does that mean? A lot of edema. Why this is inhibiting phagocytes? He was like, okay, so the phagocytes come to kill it, but when it tries to enter, it's too hard for it to enter because there is a lot of fluid here, thick fluid from the cyclic AMP, so it cannot basically penetrate that. So it gets stuck and it cannot phagocytose it. It's like, okay, that is actually interesting. So, so as you can see, we have the Bordetella pertussis, which is the leading cause of the whooping cough. So it can lead to whooping cough. Please know that the whooping cough, actually the cough, it's after you, when you take back the breath in, you do something like this. This inspiration, this form of inspiration, really deep inspiration, that is the whooping cough. That's what we are looking for, okay? So, child cough on expiration and whoops in inspiration. So it's like, so they also call it the 100-day cough, especially in China. And why? Because it can lead to a cough that can last for 100 days. So it's associated with post-tussive emesis. So the causes of Bordetella pertussis, please know what it is. It's a bacteria. So if it is a bacteria, can you treat it? Can you treat it if it's a bacteria? Yeah, you just give antibiotic and you can get rid of it. Azithromycin. So the next one is inhibitor of neurotransmitters like the Clostridium tetani and the Clostridium Battalionism. So the Clostridium tetani lead to, it has this toxin called titanospasmin. And this titanospasmin leads to spasticity. The way they work at something called a snare protein. So that's what I want to show you. So, yep. So they work at the presynaptic interneurons, which inhibit the release of GABA and glycine. GABA and glycine are neuroinhibitory neurotransmitters. So if they are not released, the patient will get spasms. Why? Because his further neuron is highly active and it's not inhibited. Okay? Why? Because the presynaptic cleft did not release the GABA and the glycine. Okay? So you can see here the following. They mentioned titinoplasmin. Both are protease that cleaves the snare. Soluble NFS attachment protein receptor. So what is the snare? Snare is a protein, basically a presynaptic protein that is located in the presynaptic cleft that helps the movement of the vesicles to where? To the cleft itself. So if the snare protein is inactive, you will not have movement to the presynaptic cleft, which means that basically you will not have passage of the neurotransmitter to the cleft. Which neurotransmitter? The inhibitory one, which are glycine and GABA. Glycine and GABA. So take a look here. So a set of proteins required for neurotransmitter release via vesicle fusion. So this can be in two cases, in the Clostridium teteri or botulinism. The only difference between these two is that the titinoplasmin works at the inhibitory neurotransmitters, while in Clostridium botulinism it works at the stimulatory neurotransmitter. So in tetany, the toxin prevents the release of GABA and glycine neurotransmitter from the ranch cells in the spinal cords, where in the something called the interneurons, okay? The interneurons, which are an inhibitory neurons. That can lead to spastic paralysis, rhesus sardonicus. This is interesting. Take a look at it. When I take a look at that, it reminds me when I got my score for step 1. Yep. Enjoy. Have sweet dreams. That's fake. But for real, they get something like this. Okay? They have really stiffening of all of their body. Okay? What did I send? Okay, that was accidental. Okay, so basically I just wanted to show you this. This is Rhesus sardonicus. And yeah, the next thing is a lockjaw, so they cannot bite. And there is one more thing called opisthotonus, which is basically a backward arch of your back because of the spasticity. So if you take a look at this patient, they look like this. Oh yeah. Good. So when we speak about the Clostridium botulinism, in the other hand, it's can be in either infant or old people. So write a note. In infant, it is because of the spores. In adults, it's because of the toxin. Again, infant, spores. Adults, toxin. So infant botulinism caused by ingestion of the spores from the soil or raw honey. So this is why honey is not allowed for children. Yep. So if you have this old ant that is like Oh, let's feed this poor child some honey so he will become healthy. Tell her to fuck off. Because if you feed that child honey, 18% of all honey in the world have endospores. So a question comes. Why, when we eat honey and it has a Clostridium Botulinism, nothing happens to us? No, we don't have immunity. Well, here is the thing. We beautiful adults, we have bacteria in our colon. Yes? We have a lot of bacteria in our colon. So when we get the Clostridium Botulinism spores inside of our colon, does it have a place to germinate these spores and make bacteria out of it? Does the Clostridium Botulinism have a place to germinate inside of our colon, or we already have an occupied space? There is no way it can germinate. We already have no space for it, yeah? So it will not germinate. But if we speak about a baby intestine, they are sterile, like their souls. So it's really sterile. There is nothing inside of it. So if you get the spores, they can come here and be like, oh, it's so clean. I love it. Let me germinate here and start producing the toxin. For the adults, you don't need the spores. Because if you get the spores, nothing happens to you, basically. But if you get the toxin, something happens to you. So where you can get the toxin from? From home canned food. So the canned food, please be careful actually, this is important. When you buy food at the supermarket and you take a look at the can and it's a little bit puffy, don't take that one. Don't take it. Even if they write, oh, we will make you really good discount in the buffy ones. Yeah, because they give you an extra Clostridium Botulinism. You don't want that. So, don't take it. That's the take home. Okay, so that's about, in general, about the Clostridium Botulinism. It can lead to a floppy baby syndrome, in which the baby normally is flexed. Babies are flexed. But the floppy baby, they will look like a tissue. Basically, they will be, see? Baby normally should not be like this. Baby should be active. He should be always flexed. So when he's like this, he's a floppy. That's a sign of a floppy baby syndrome, which only exists in two diseases that we'll speak about in the USMLE. One of them you just saw, it was the Clostridium Botulinism. So the next thing I'm going to speak about is no, the other one is not actually Guillain-Barré. Guillain-Barré is an ascending paralysis. I'm speaking about a total paralysis that affects all over the body. Well, that's anterior muscular dystrophy, spinal muscular dystrophy. That we'll study that in neurology. It's due to sublysosome defects, which we'll study in biochemistry. So that's for later. So, light cell membranes. Some toxin can destroy the cell membranes. For example, the Clostridium perfringens can destroy the cell membranes using something called the alpha toxin. When you do sketchy, you will see that. Remember, our cells have phospholipid bilayer around them. So when we speak about the alpha toxin, it's basically a lecithinase, which is what? What is a lecithinase? It's a phospholipase that degrades the cell membrane, which can lead to an horrible myonecrosis, okay? And gas gangrene and hemolysis, double zone of hemolysis on the blood agar. I actually have to show you a photo of myonecrosis just for you guys to appreciate the seriousness of this disease. So this bacteria produces gas, and this is important. So you can see this patient, he has myonecrosis. The next step is actually to open his skin, because if you leave it a little bit more, it will lead to compartment syndrome and he will die. So this thing done here, the cut, are they from the bacteria or the doctor did them? The doctor did them. Doctor did them. Yep. Why he did them? Because He wanted to expose it to oxygen so it will die because it's an obligate anaerobe. Yeah? So that's important. But myonecrosis, I'm seeing only horses. I just need to see a human. Okay. So I want you to know one thing. Guys, this produces a lot of gas. Yeah? They produce a lot of gas inside of them. So you can take a look here. You can see a little air buckets inside of here. But this is not good enough. Oh, this is a good one. Okay, you can see little air buckets here. Yeah? These air buckets are important. Why? Because if you see this, this is an indication of surgery. If you take a look here, this area, this is an air bucket. Guys, when you press on it, it's like stepping on the sands. It will make a sound like which is a crepitus. Yeah? That's a crepitus. It's like you hear that sounds, done. That's gases under the skin. So what you need to do, open it, cut him right away. So, Strep Biogenesis. It makes something called the streptolysin O, which is a protein that degrades the cell membrane as well. So it lyses the RBCs and contribute to the beta hemolysis when you guys will study the Streptococcus pyogenes. It hosts antibodies against the toxin used for diagnosis of rheumatic fever. Please do not confuse it with immune complexes of post-streptococcal glomerulonephritis. So, if you remember, guys, we spoke about post-streptococcal glomerulonephritis. That was a type 3 hypersensitivity reaction. It's completely different. Yeah? But when we speak about the streptolysin, this is just a toxin that can lead to beta hemolysis on the agar itself. I mean, on the culture. And it can be used also as a marker for rheumatic fever. That's it. Rheumatic fever is not the same as post-streptococcal glomerulonephritis. Post-streptococcal glomerulonephritis is a form of nephritic syndrome that happened as a complication of the strep throat. Rheumatic fever is a cardiological disorder, cardiovascular disorder. If you remember, it had the Jones criteria, in which they had the joint stuff, which was the arthritis, the heart stuff, the pancreatitis, the nodules under their skin, and they had also the erythema marginatum and the Sydenham chorea. Okay, so the last thing is super antigen causing shock. So, speaking about the super antigen. Super antigen can be in case of Staphylococcus aureus or Strep Biogenesis. Basically, what I want you to know about this super antigen, that they are super and they are antigen. Kidding. Let me show you exactly what they do, because it's really important. Normally, you have the antigen presented on the MHC class 2. Yes? So you normally have this MHC class 2, and you have the T cell receptor, and you have the antigen in between. Yes? And this is the antigen presenting cells here, of course, and this is the T cell, of course. So this is the T cell, and this is the antigen presenting cells. What occurs in this case, that they cross-link the beta region of the T cell receptor. You are like, okay, what is the T region? Well, see this, guys? This is not the T region. The T region is here, okay? So the toxin, what I'm trying to say, that the toxin does not bind to the area that it was supposed to bind. I'm going to make the toxin with the green. So the toxin instead binds to this area, okay? So they write, it cross-links beta region of the T cell receptor to the MHC class 2 receptor on the antigen presenting cells outside of the antigen binding site. It does not bind to the antigen binding site. It leads to direct activation of the T cells. So when the toxins lead to direct activation of the T cells, what that will lead? It will lead to overwhelming release of cytokines, including interleukin 1, interleukin 2, interferon gamma, and TNF alpha, which basically lead to a shock in the patient. Okay? Exactly, cytokine storm, kinda. Do not confuse it with the cytokine storm in COVID, because that's much worse, and basically it's mainly mediated by interleukin 6, not 1 and 2. If you ask if we will discuss, do you think, guys, we will discuss COVID in USMLE? It's so new. Will we be discussing it? Well, that was not a good answer. I did not like that. So we will. We will. There is a whole page in first aid about it. There is a whole page in first aid about it, in which we will speak about every single pathology it can lead to, from the cytokine storm until how it kills, how it harms the patient. Just want to show you the page so you will not be surprised when you see it. Okay. Yep, can you see severe acute respiratory syndrome, coronavirus 2? Yep, that is COVID. This is COVID, COVID-19 virus. A whole page about it. We'll speak plenty about this. Okay, good. So come on, guys. One last thing and we can call it off. So you can see here that toxic shock syndrome can lead to fever, rash, and shock. Other toxin can lead to scaled skin syndrome, which is an oxyphiliative toxin, and food poisoning, a heat-stable enterotoxin. All of these can come from the Staph aureus. Basically, the Staph aureus can lead to this scaled skin syndrome. You must take a look at that. And I want to ask you one question about it. When you take a look at this, guys, if you put your hand on this patient, okay? And then you push it against his body, will his skin slough off? Yes or no? Yes, it will slough off. That's Nikolevsky sign positive. Basically, these poor babies, when you put your hand on their skin and then you move it really strong, their whole skin sloughs off. And that's a positive sign of Nikolevsky. Nikolevsky positive sign. Okay? So, okay. So also this Staph aureus can have an enterotoxin, which can lead to really bad vomiting and diarrhea. But I want you to know something about this one. The enterotoxin of Staph aureus can lead to diarrhea and food poisoning, but it occurs really quickly and disappears really quickly. Okay? So toxic shock-like syndrome can lead to fever, rash, shock, and scarlet fever. That's in Strep Biogenesis, not Staph aureus, because it's not the real deal, basically. Okay, so in this one, you will not have a scaled skin. That's the difference. For now, I want to call it off. Tomorrow, when we come, we finish off with endotoxins. We'll be starting clinical bacteriology. We'll study alpha hemolytic, beta hemolytic. We'll speak about Staph aureus, Staph epidermidis, Sabrophyticus. I want you guys, please, to do strep pneumonia and Virulence and group A and B. Also, try to do Enterococcus and try to push as much as you can with the gram-positive road. You should not finish all of them, but as much as you finish, guys, the easier you will make our work goes, the faster we goes. So try to watch as many videos as you can today. I sent you the link. One of our great students sent it to me, so that this way it's actually much more easier to see the videos than the link I sent. The link I sent, it was compressed in RAR. This videos are not compressed in RAR, which makes it more convenient and more easy for you guys to use. Until tomorrow, I will see you guys. Please don't forget to watch the videos, because without them, it will be really difficult class tomorrow. With them, it will be super easy and fun class tomorrow. So see you guys tomorrow.